Welcome and thank you everyone for joining us for our webinar presented by Protara Therapeutics titled The Importance of Choline for People on TPN. Sharing a couple of housekeeping items before we get started. This is a webinar format, which means your camera and microphone will be off and we'll only be able to see the presenters. While we will not be utilizing chat or the raise hand feature, you can use the Q&A function in the toolbar at the bottom of your screen if you have questions during the presentation. And our presenters will answer them at the end as time allows. This webinar is recorded and it's going to be available on our website at oley.org/webinars. The information provided during this and all of Oley's presentations is for educational purposes only and is not intended as medical advice. Although clinical information is peer-reviewed, you are advised to consult with your healthcare team before making any changes to your care plan. To get us started, please allow me to introduce your speakers today. Dr. Alan Buchman is a gastroenterologist, professor of surgery and medical director of the Intestinal Rehabilitation and Transplant Center at the University of Illinois. Dr. Jacqueline Zummo is co-founder and chief scientific operations officer of Protara Therapeutics. Without further ado, I'll turn it over to you, Dr. Buchman and Dr. Zummo.
Thank you very much, and good morning or good afternoon to everybody. There are four goals that we have in our presentation today, and these are, number one, to learn about choline and why it's important for people that are on parenteral nutrition. Choline is not really a vitamin, although it's similar to the B vitamins and is often included in that class, but it's actually what's called a quaternary amine, which doesn't mean anything to you, but it's vitamin-like. And we'll learn today why choline is important for individuals that are on parenteral nutrition. I will talk about some of the clinical trials that we've done to look at choline deficiency and supplementation of choline in patients, consumers that receive parenteral nutrition. We'll also Jackie Zummo will speak about some of the more recent trials that have been done and an interventional trial with giving IV choline that is just starting. Lastly, there have been actually some questions that were already submitted, which we'll answer, and if time permits, we'll try to answer any other questions. And if time doesn't permit, I would encourage you to send any questions to Oley, and they will send them to Jackie and myself, and we'll do our best to answer them for you. So what is choline and why is it important? Well, choline is a source of what are called methyl groups, which is a carbon and three hydrogens. And these are really important groups because they're used for repair of DNA. And when DNA can't be repaired, that's one of the major risk factors for developing cancers, for example. The biggest role of choline is that it's present in the cell membranes of every cell in the body, usually via a moiety called phosphatidylcholine, of which choline is the main constituent. It's very important in order to maintain the integrity of the cell walls. Choline is also the precursor for a neurotransmitter called acetylcholine, which is the way nerves communicate with each other. And that's, of course, necessary for memory, to be able to move your arms and legs, your heart function. And in addition, actually, more recent evidence suggests that acetylcholine is actually necessary for normal bone metabolism as well. One of the most important roles of choline is for the liver. In fact, choline is synthesized in the liver, and we'll talk about that, but there's a problem, and the reason that consumers on parenteral nutrition develop choline deficiency is because the liver isn't able to synthesize choline, because there's a difference whenever you eat nutrients, they're absorbed in the GI tract. Usually most nutrients are absorbed in the very beginning part of the intestine, and then they go to the liver, where the liver metabolizes them to other nutrients. So for example, with amino acids, there are what we call essential amino acids and non-essential amino acids. Well, you still need the non-essential amino acids, but the liver is able to take the essential amino acids from the diet and make them into the non-essential amino acids. And that's one of the reasons that choline was not thought to be required for humans, because the amino acid methionine is included in your parenteral nutrition. And when the methionine is taken in the diet, the liver is able to metabolize that methionine to choline, among other products. But that doesn't happen in consumers that are on parenteral nutrition. And so you have to obtain choline from the diet. But the problem is that if you could get enough choline from your diet, well, you wouldn't need parenteral nutrition because all the nutrients and fluid are malabsorbed. The other problem with taking a lot of choline in your diet is that choline is metabolized by bacteria that are normally present in the beginning part of the bowel to trimethylamines. And trimethylamines is the substance that's in bacon, for example, that is a recognized carcinogen. And so taking too much choline in the diet, trying to overcome malabsorption of choline, is not a good thing either. So currently, obviously, none of the parenteral nutrition products have choline, except there's a tiny bit in most of the lipid emulsions, but not nearly sufficient to meet needs. About 80% of people consumers that receive parenteral nutrition, according to a study that I did with about 65 patients and another study that Charlene Comfort did from the University of Pennsylvania, will become choline deficient. The most prevalent finding is that a fatty liver develops. And a fatty liver by itself may not be a bad thing, but when other factors come into play, it can lead to very rapid progression of liver disease. There is also some evidence, which I'll discuss as well, in terms of some memory impairments that may be improved with choline. And studies in normal volunteers, not consumers on parenteral nutrition, have even shown that if they're fed a diet that's deficient in choline, they develop muscle damage. And the enzyme creatinine phosphokinase, or CPK, is released into the blood, and this indicates muscle damage. And some studies that we have done also show that there's a substantial risk for developing catheter thrombosis when you're choline deficient as well. And I'll show you some information on that. In normal volunteers that we put actually on parenteral nutrition to study what happens to the GI tract, we also actually measured choline concentrations in the blood. And within a week or two, these decrease by about a third or even half of normal, although none of the patients actually developed choline deficiency. In another normal volunteer study done at the University of North Carolina by Dr. Steven Zeisel, he put normal college students, I don't know how many of them are really normal when they participate in a study like this, but they were given a choline deficient diet, and they developed liver test abnormalities to the point where the study had to be stopped prematurely. Now, choline, the big issue with choline is that the deficiency of it drives liver injury in consumers that receive parenteral nutrition. And the problem here is that although there is an interaction between choline and vitamin B12 and folate, there's the folic acid and vitamin B12 are unable to compensate for a complete deficiency of choline. And choline is required to produce the cholesterol called VLDL, or very low density lipoprotein. And that cholesterol is actually very important because it transports fat out of the liver. And so when there's a deficiency in VLD or deficiency in choline, there's a deficiency in VLDL, and fat sort of backs up in the liver. The other thing that happens with choline deficiency is there's a decrease in the transit of bile from the liver to the gallbladder where it's stored, and bile can back up into the liver, and you get cholestasis. And some of these bile salts can be toxic to the liver cells. Now, in terms of biopsies, there's a couple different findings that we find in consumers that have liver disease related to intestinal failure. We call that intestinal failure associated liver disease. The first one is steatosis, and you can see all this white, or these are all fat globules. And one of the interesting things about intestinal failure associated liver disease is that there's a fairly unique appearance in that some of the, you can't really appreciate it on this particular slide, but some of the cells are big. We call that macrosteatosis, and some of them are very tiny, called microsteatosis. And there's only a handful of diseases that have a histologic appearance like that, such as like Jamaican vomiting sickness and a couple of others. Cholestasis, and this is actually what predominates in adult patients, although not in all adult patients. In preterm infants, as well as a few adult patients, cholestasis is what is predominant. And you'll see a little bit, it's hard to see on the slides, but you see a little bit of yellow pigment here in the biopsy. And that's because, again, the bile flow is reduced or blocked from choline deficiency. The other thing is that there's an increase in apoptosis, which is programmed cell death. And this has been shown not only in liver cells, but also in lymphocytes, a particular type of blood cell that are important to fight off infections. If you put a rat on a choline deficient diet for a year or year and a half, some of them will develop actually liver cancer or hepatocellular carcinoma. Now, in this study from Paris, from Bernard Messing's group, chronic cholestasis, which was measured by liver test abnormalities, the ALT, AST, and alkaline phosphatase, occurred in almost over 70% of their patients after about 6 years of being on parenteral nutrition. We don't see the prevalence quite so much these days, which is in part due to just better overall care, better care of infections, the new lipid emulsions, but it still does occur. And I will caution people that even with the new lipid emulsions, your numbers may get better, but your biopsies don't. And patients still go on to need intestinal transplants or intestine liver transplants. And in fact, intestinal failure associated liver disease is the leading indication for an isolated bowel transplant or a combined bowel liver transplant. In terms of more serious disease, of course, in this study, after 8 years, this is again from the French study, over half of their patients actually really had quite significant liver disease. I will point out, though, that not all patients develop cirrhosis. It is possible to develop liver failure in the absence of cirrhosis and still require a transplant. And that's really what we want to prevent. We want to prevent any of this liver disease. As I mentioned earlier, choline also plays a role as a precursor to acetylcholine, which is the most important neurotransmitter in the body. And there are a number of studies actually that have been done in Alzheimer's disease, in tardive dyskinesia, Huntington's chorea, and Parkinson's that have looked at supplementation of choline in terms of treatment of these diseases. The data is a little bit plus minusy with some studies supporting the role of supplemental choline and others not. The point that I'll make, though, is that none of these patients were actually choline deficient, and the choline was not being used to replace choline that would normally be present in their diet, but being used as a supplement. And as I mentioned, if you take too much choline by mouth, you can also have other problems. There's been a link to developing cardiovascular disease and an increased risk of stroke. And that, again, is because of the metabolism of the choline in the diet to trimethylamine and trimethylamine N oxide, which does not occur in when choline is administered intravenously. Now, in a small study that we did, with taking consumers on parenteral nutrition and giving them a placebo or giving them choline, you can see here that we significantly elevated the plasma choline concentration. And there are also some improvements in both visual and verbal memory as measured by the Wechsler memory scale and the California Verbal Learning Test, as well as the TRAILS-A test. The TRAILS-A test is an examination where the individual connects dots, connects numbers or letters to each other. So there is some preliminary data that when one is choline deficient and that choline is replaced, not as a super physiologic situation, but just replacing that choline so that choline deficiency is reversed, there may be some modest improvements in verbal and visual memory. As I mentioned, choline also plays a role in muscle health. The normal volunteer study from the University of North Carolina showed that muscle damage occurs when choline is removed from the diet. And again, that probably relates to the fact that choline is an integral part of the cell membranes of all the muscle cells. There's also an increased risk for catheter thrombosis, and obviously there are quite a number of different reasons that thrombosis can occur. The most important of which is malposition of the catheter and a vein that's too small, for example. But in our study, we found that in our patients that if you had a choline level that was less than 8 nanomoles per mil, which would be considered abnormal, there is a tenfold increased risk of developing catheter thrombosis, irrespective of other risk factors as well. And as I mentioned, choline also has an important impact on bone health. We know that consumers on parenteral nutrition will develop sometimes osteomalacia, sometimes a low turnover osteoporosis, or a combination of both. And we've never really figured out what the problem is. Some of it's related to the underlying disease as well as aging. But more recent data has shown that neurotransmitter acetylcholine has an important role in in the development of bone density, increase in bone density, and by stimulating bone growth. And as I mentioned, in patients that are choline deficient, acetylcholine may become deficient as well. And this is something that I think will need to be looked at in consumers that receive parenteral nutrition, whether bone density can be improved and the metabolic bone disease that consumers get, whether that can be reversed by providing choline intravenously. Now, in terms of replacement guidelines, Aspen as well as Espen have also both recommended choline in parenteral nutrition, but there's one problem, and that problem is there isn't such a product. And that's what we're here today to talk partly about. So I'm going to review some of the early clinical studies that we've done with intravenous choline in consumers that have received parenteral nutrition. A few definitions so that you understand studies better. A single blind study means that there's no placebo, and so the patients and the doctor or one of them knows what treatment they're getting. So sometimes if a patient knows or a consumer knows what treatment they're getting, they may think that they're getting better. In fact, actually, in the memory study, we had one individual who's still with us who thought that they were getting smarter when they received choline, except when we broke the code later, we found out that they got placebo. And so that's one of the reasons that we want to use a double-blind study, so that neither the doctors who can influence the patients or the subjects and consumers themselves know what they're getting. But what happens here is that we don't pick and choose. We just randomize patients, which means that we don't know who's going to get the drug and who's going to get the fake drug. An open labels trial, on the other hand, is where everybody knows who's getting what drug. And sometimes that's useful actually from a safety standpoint, because we don't really care what the incidence of problems is with a placebo, but we certainly care whether there are problems that develop with the drug, or in this case, choline. And of course, obviously, you'll interact if you're in the study with a principal investigator, which is a physician, or the site coordinator, which may be a nurse or a PhD or a dietitian or other. So initially, we started our work in the early 1990s. I actually had looked at this when I worked with Dr. Zarrao and Lombardi at the University of Pittsburgh in the 1980s. And when I was getting my master's work, we had to do a paper on two nutrients from the big Shills and Young nutrition textbook. And I was working as a full-time fellow, and so I picked the shortest chapters, which were choline and carnitine, and found that the carnitine really didn't do anything for patients that were on parenteral nutrition. But there were a lot of animal studies that suggested choline deficiency. In fact, about a dozen different animals, from dogs, baboons, chickens, trout, where these animals all developed fatty livers when they were fed choline deficient diets. And so we first fed a lecithin diet because there was no IV choline. This lecithin was about 19% choline. We gave a very high dose, just short of what would cause nausea and vomiting. And I'll show you, well, actually, I don't think we have the slides in here, but the bottom line is that we were able to decrease the fatty liver and increase choline concentrations in the blood, but this fatty liver remained, and the choline levels were still below normal because we just couldn't give enough. And so in 1994, we did our first trial with 4 patients with choline that we made up in the lab to find out, well, what seems to be the appropriate dose, which was 2 grams of choline chloride, that's not choline, it's choline chloride. The DRI, daily recommended intake, is about 550 milligrams per day in the diet, and we're pretty close to that. And I'll talk a little bit about the phase 2A trial that we did and published in 2001 in JPEN. And one of the earlier studies that we did also is we took many of our patients and we looked at their choline concentrations, and we looked at the correlation between the liver test abnormalities that you're familiar with. And the bottom line is that the higher the choline level, the lower or even normal the ALT and the AST. And there was also a correlation when we did CT scans of the amount of fat in the liver with the choline level. So the higher the choline level, like here, there are 2 patients where it was normal, and they don't really have any fat in their liver. And these patients, of course, did when they had lower choline levels, and the lower the choline level, the more fat in the liver. So in this placebo-controlled trial, and again, the red representing consumers that receive choline in their parenteral nutrition, and the blue representing those that receive placebo, when we look at the choline concentrations, of course, it quickly goes to normal. Normal is about 10 to 15 nanomoles per mil. Obviously, some people got way above normal, but toxicity does not occur unless it's over about 200 nanomoles per mil. When we look at a marker of cholestasis, which is the alkaline phosphatase, we also found that there were significant improvements, and you can see the breakdown here between those that got the placebo, continued with elevated alkaline phosphatase, and those that received the choline. There were substantial decreases, 20 to 30% of their abnormal alkaline phosphatase. And when we look at the liver, this is an example of a consumer with a very fatty liver, and you can see the liver here, and this is the spleen, and these should really be the same density. And after they received a month of, actually 4 months of choline, you see that there's still a little bit of fatty liver here, but it's pretty much the same as the spleen in terms of density. We don't use CT so much anymore. It's more MRI because it's newer and more expensive. And so the CT results were converted to MRI, and we see the same results where liver density significantly decreased as the fat decreased in the liver with the group that received placebo, and remained unchanged in the group that received the placebo. So the bottom line is that the IV choline was shown to be pretty safe and well tolerated, although, again, very small studies. There were some serious adverse events in the study, including a hospitalization for a patient for an infection, but that was a patient that received placebo, and none of these adverse events were attributed to choline. There were actually more infections in the group that received placebo, not enough to really make much out of, but there is some suggestion from some studies that individuals that are choline deficient may have some immune deficiency. Okay, so with that, I'm going to turn the slides over to Dr. Zummo, who's going to talk about the current study that is just underway of IV choline in consumers. Jackie?
Sure. Thanks, Alan. Next slide, please. First and foremost, I think it takes a minute to just introduce everyone to Protara. It's worth it. So we're Protara. We're a small company, a little less than 50 individuals. We work out of New York City. That's where our headquarters is located. We were founded in 2017 by myself and our co-founder, Jesse Shefferman. Really, what we look to do is try to prioritize creativity, having a diverse perspective, and really just don't give up on our goal of bringing therapies that can change the lives of patients to those patients. Next slide. So we've been working with the FDA since 2017 on IV choline chloride. And what I mean by working with them is that we have gone through multiple rounds of discussion with them about what's the appropriate way for choline to be added to parenteral nutrition. What is the clinical trial design that needs to be undertaken to be able to bring this medication to patients who are receiving parenteral nutrition? And through that process, we've arrived at where we are today, which is with a study that the agency has agreed is appropriately designed to be able to lead to the registration or the commercialization of this product for patients. Through that process, we also were able to receive fast track designation. And that designation is a nod from the agency to basically highlight the importance of this product. And so we are really excited about that because it leads to our ability to be able to bring this product to patients faster than the normal typical review process and timeline that the FDA takes for programs that are not designated in this way. Next slide, Alan. So we have, over the course of the last 7 years, conducted a trial that led to our ability to further understand choline deficiency in this patient population. And as Alan mentioned, he had conducted his trial back in the late 90s, early 2000s, and we really wanted to be able to demonstrate to the FDA that there was still a need for choline for those that are dependent on parenteral nutrition. So we undertook an observational study. That was Thrive 1. We ran it globally around the world. And what we found was that approximately 80% of patients who were receiving parenteral nutrition were in fact choline deficient. And for us, that was really important because what we also learned was that those patients, about 64% of those that were choline deficient, also had some sort of liver dysfunction. And that really tied to the data that Alan had generated in his placebo-controlled study and gave us the opportunity to go back to the FDA and have a conversation around what our registrational phase 3 trial could look like and what we really needed to be able to measure to demonstrate to the FDA the value of choline in this patient population. So that leads us to Thrive 3, which is the phase 2b/3 trial that we are currently conducting. It's going to be both in the US as well as around the world. We now have approval from the FDA on the design. We've opened our first clinical trial site. We'll continue to open up clinical trial sites. Next slide, Alan. So as I mentioned, Thrive 1 was really important for us to demonstrate to the agency that choline deficiency was really still something that was happening in this patient population. And, Alan mentioned earlier that there had been some changes in the availability of different types of lipids, medical management of patients who were dependent on parenteral nutrition. But really, at the end of the day, even several, decade after what Alan had done in his research, it was determined that still choline deficiency was still pretty prominent in this patient population, and that liver injury was still, again, pretty prominent in that patient population. Next slide. So this is a clinical trial, and I think that it's really important for us as Protara, as the sponsor, to explain what it means to be part of a clinical trial and what a clinical trial means in the long run, both for patients that are in the trial as well as for the patient population from just a broader sense. Clinical trials require people to come into them to be willing to undertake the commitment to be part of the trial and to commit to being able to help us to determine the benefit and the risks associated with the product that we are trying to bring to market and to make commercially available. So, we look at clinical trials to determine the safety, the effectiveness, as Alan mentioned and walked you through with his earlier trial. This trial is going to be much larger than the trial that Alan ran. And the reason for that is because we really want to make sure that we have an appropriate safety understanding as well as efficacy understanding, and that it's generalizable to the total population that will eventually receive this product. Next slide. So our ability to conduct these trials really relies on participants' willingness to come into them. And we value that participation more than anything else, because it really is an individual's commitment to their own healthcare as well as to the healthcare of the broader patient population. We really couldn't do this without all of you. So we are trying and have been able to align with the agency on an eligibility criteria that allows for the broadest patient population to be able to come into our trial. And that's really important because it gives the largest number of patients who are receiving parenteral nutrition the opportunity to actually have the ability to determine whether they're choline deficient, to be able to receive the product. And in the case of our study, receive the product until the drug becomes approved. Next slide. So when you're thinking about being part of a clinical trial, there are obviously the steps that you need to sort of consider. The first is just considering your options, whether or not the trial is right for you. It is a commitment. You are expected to be able to adhere to the protocol, and that means coming to see your doctor maybe a little bit more frequently than would be the case if you weren't part of the trial. It's also, understanding the risks and benefits of being part of the clinical trial and making a commitment to being able to really be part of it. And at the end of the day, it really is also about the screening process, right? As I mentioned, we did work with the FDA to ensure that the broadest patient population would be able to come into this trial. And so for us, we think that that's really important, and it leads to the ability for, generalizability across the patient population. Next slide. Alan talked to you a little bit about what a placebo is. And in the case of this trial, the phase 3 component of it is a placebo-controlled trial. It gives us the best opportunity to be able to demonstrate to healthcare providers as well as to the FDA the objective benefit of our product versus a placebo, which Alan mentioned is not an active component. Next slide. So the Thrive-3 clinical trial, as I mentioned, is the registrational study that is going to be conducted in patients who are receiving long-term parenteral nutrition. And what that is for us is the demonstration that the agency has recognized the unmet need. It also, as I mentioned, has fast track designation. It is the only study that will be required for the approval of this product. And we're hoping to dose our first patient in the next month. As I mentioned, we have our first clinical trial site open, and we're looking forward to being able to start the action of actually treating patients. Next slide. So our patient population is adolescents and adults. Adolescents defined as 12 to 17, adults 18 and older. The main criteria for inclusion into this trial, or the ability to enroll into this trial, is that an individual needs to be receiving parenteral nutrition at least 3 days a week, and they need to have been on parenteral nutrition for 6 months or more and expected to continue to receive parenteral nutrition. The design of the study is 2 parts. The first is a 4-week open label dose confirmation phase of the study, which will give us the opportunity to test 3 doses of choline, pick 2 to move forward into then the 24-week double-blind placebo-controlled study that will then go out to 24 weeks. And both of those components of the trial, both those cohorts, will be able to continue into an open label extension that will continue to provide IV choline chloride to all patients until the product is approved. Our primary endpoint is determining PK, and that really just means the levels of choline in the blood. Our secondary endpoints are, of course, to look at liver function, but also to look at some of the other potential downstream consequences of choline deficiency that Alan talked to you about earlier. Next slide. So this is the overview. Again, it's really about dose selection, moving into being able to confirm the doses, and then going into looking at the RCT to determine safety and effectiveness. We've got several sites that are going to be opening around the world. A lot of them are going to be in the US. We're really looking forward to having, the Oley membership be part of this trial. Next slide. As I mentioned, our inclusion and exclusion criteria are as broad as we possibly could have it, mainly because we wanted to be able to give all patients an opportunity to come into this study. Next slide. We have posted on clinicaltrials.gov a clinical trial listing that gives more in-depth information in terms of the detailed inclusion and exclusion criteria. We also are, if you go there, you can see the sites that are activated in the U.S. And as new sites get added, there will be new additions to that webpage. If you're interested in participating in this trial, we do have these websites here, or excuse me, these email addresses here that you can reach out directly to the Protara team for us to be able to direct you to which locations could potentially be of potential option for you to come into the trial. The other thing to know is that we do try to make it easier on patients in terms of being able to be part of the study. So there are actually, ways for us to ensure that if you have to travel, that we can help with that. We encourage everyone to spread the word about this study. Talk to your healthcare providers about it. Talk to other, friends and other people that are dependent on parental nutrition and that are receiving parental nutrition. Next slide. This is our current map of our clinical trial sites. These are the sites that have been selected and that will become open and active and able to enroll patients, either have already been activated or will become activated over the next couple months. We are looking to expand this number of sites and also to try to have a better distribution across the US. But for now, these are the ones that we have selected. Next slide. That's the end of the presentation. Thank you.
Thank you so much, Dr. Zummo, and thank you, Dr. Buchman. We really appreciate the information and this great time where we can address a lot of the questions that were submitted. And if any of you listening have a question, please feel free to submit in the Q&A feature, and I will take a look at those now. And John and Melissa, if you are on, if you can just let me know. Let's see, I think they're popping in right now. Okay, perfect. So we are going back to the beginning because we have a lot of questions in here. Okay, so the first question, thank you everybody for submitting, is how can I participate in the Thrive 3 choline clinical trial? And I think Jackie may have just touched on that, but that was the first question to come in.
So how to participate. So you saw the sites that are currently selected for the trial. I think the best thing to do is to talk to your healthcare provider first and foremost. Then they can reach out to Protara, also to within the network in terms of the PIs that are listed to be able to potentially figure out how best to manage transition into a site where we actually are active and open and enrolling.
Perfect. Thank you, Jackie. And the next question, this may be for you, Alan. It's what studies were done to ensure choline can be safely added to the parenteral nutrition bag?
Well, there have been a variety of studies that have been done. We had done some, and Protara has done others. There have choline in various concentrations has been added to parenteral nutrition solutions, various solutions with different concentrations of amino acids, of protein, lipids, with lipids, without lipids, to look at stability, to look at any changes to the solution itself, and then obviously in the clinical trials. One of the main things that we'll be looking for are whether there are any unanticipated side effects from choline. So participants in the study will, of course, be queried about different things. They'll have examinations, so on and so forth. The important thing to recognize is that we're really giving in the study replacement doses of choline. So it is technically a drug because it's administered intravenously, but it's really not much different from like giving vitamin B12, or giving glutathione, or anything else that's maybe in the parenteral nutrition solution. But nevertheless, it's important that we look for side effects, and of course we'll look at the blood levels in anybody that does develop a side effect. When we did our initial studies, we gave some very large doses of choline, much greater than what will be used in this study, to see whether we could induce side effects and what those were. And in one patient that we were able to give, who had kidney problems, and we were able to give like 4 times the amount of choline that you'd get in the study, and got her levels to over 200 nanomoles per mil. She developed some sweating and a little headache, and some abdominal cramping that resolved when it was stopped. It was pretty minor, and those are levels that are 15 times what's normal. So, but we'll be looking for all those things, as well as the potential for other things that we hadn't seen previously.
Thank you, Alan. And the next question that came in regarding the PN, I actually had this question myself as a nutrition support dietitian. How is choline added to the PN bag?
Jackie, you want to?
Yeah, sure. So in the clinical trial, we actually tried to be as flexible as possible, and it can be added either at the time that the parenteral nutrition is mixed initially by the home health pharmacy, or it can be added by the patient right before the infusion starts.
Okay, perfect. And a question that kind of piggybacks off that a few people had is, how does someone know if they are choline deficient?
So right now, there is no assay commercially available to be able to determine choline deficiency. So the only way would be able to be coming into our trial.
Gotcha. Okay. And then if someone's not sure if they're deficient and they enter the trial, how can they tell if choline is helping them?
So there will be visits that are throughout the trial that choline will be measured, and in the open label phase 2B component, because it's open label, we'll know right away. In the randomized controlled study, we won't know, or we won't be able to say specifically until the trial is done.
And there's a variety of different things that'll be done. I mean, we'll be looking at grip strength to see if muscle strength improves. We'll be looking at the choline levels. We'll be looking at the fat in the liver. We'll be looking at the incidence of infections and thrombosis. But as Jackie mentioned, it's because it's a placebo-controlled trial. We're not going to know until the trial is over who got choline and who didn't. So that way we can see whether there were differences in those things. Like, for example, did grip strength improve in those that got choline, and it didn't improve in those that didn't.
Okay, that makes sense. And then you touched on this, Alan, earlier, Dr. Buchman, of potential side effects to get it. So potential side effects, you had mentioned someone who had a kidney disease, then they had sweating and headache. Are there potential side effects to getting choline? Anything that would make someone not a candidate? Jackie or?
Yeah, the first thing I want to mention, though, is that if you Google choline, I mean, you'll see all this stuff about cardiovascular risk and so on and so forth, and that's actually with oral choline. And so we need to be clear here. People understand that as I mentioned in my part of the talk, is that when you take choline by mouth, it's the vast majority of it, over 70%, is metabolized by bacteria that are normally present in your intestine to trimethylamines and trimethylamine N oxide. And these actually are substances that are recognized carcinogens, number one, and number two, also do increase the risk for cardiovascular disease. That means like heart attacks and strokes. But that doesn't happen when you get it IV, because the bloodstream is sterile. There's no bacteria. So IV choline is not metabolized to trimethylamine. So it's actually impossible for the trimethylines to be produced. However, If you do get too much choline, as I mentioned, there are cholinergic side effects, which are the same side effects that if you take a cholinergic drug, for example. And those, again, it was just one patient, so we don't have enough information, but it was when her level was over 200 nanomoles per mil. We're looking in this study our normal is 15 to 20. If we get some patients to 30 or 40, that's better than having still remaining choline deficient, but there's a very large, a wide range of therapeutic efficacy without side effects. But then again, individual patients can be different, and so we will be carefully looking at every patient, because the important thing here is that obviously that the efficacy of the drug outweighs any potential side effects. So we'll see. We look for things that are not anticipated. Jackie, do you have any comments on that, or?
No, that's right. I agree with everything you said.
Thank you.
And
Yes.
Tracy, you missed one in the published section of the Q&As, not the
Yes.
reviews.
Yep.
There's one down at the bottom if you want to go to the published.
Is that the 80% study?
No, it's dealing with travel costs. That's
okay.
80% studies, the review questions that are not published yet.
Gotcha. Okay, I'm scrolling back. Thank you, Melissa. Okay, my apologies. So this question came in initially. Will travel costs be covered if I participate in the trial and do not live close to one of the clinical trial medical centers?
Yes, we do provide travel cost reimbursement to ensure that patients are not restricted from participating in the trial due to travel.
But Jackie, that doesn't include flying first class on Emirates Airlines, though, correct?
Well, we prefer that it be reasonable travel, but there are reasonable budgets set forth to allow for patients to be able to get to clinical trial sites that are open and active. Our goal is to try to have as many patients be able to participate in our trial as would like to.
Okay, thank you so much. And we have one that came in. Given that choline plays a role in metabolism of B vitamins, how do mitochondrial disorders or dysfunction impact choline needs? Maybe Dr. Buchman or Dr. Zummo?
That's an Alan question.
Yeah. Actually, we don't know, actually, and we'll maybe find out if we have some of those patients in the study that may have pseudo-obstruction, for example, from mitochondrial encephalopathy or the like. B12 and folic acid are both methyl donors, and so is choline. And so in terms of DNA repair and that sort of thing, this study's not long enough to see whether we're going to prevent cancer from developing, for example, or prevent liver cancer from developing. And we don't actually, the measures of looking at programmed cell death are kind of experimental. And so we haven't planned those in this study. So that the main outcome measures, again, are going to be what are well-described clinically significant outcomes, which will include, again, the main one, can we get the choline level to normal and maintain it? We'll be looking at kidney function. We'll be looking at the liver. We'll be looking at thrombosis, infections, possibly bone density, muscle strength, all the things that either in humans or in animals have been associated with choline deficiency. But whether or not it'll improve the mitochondrial disorder itself is not clear. There's not really a good way to measure that, and there's a lot of different factors that come into play with regard to metabolism in the mitochondria.
Thank you so much, Dr. Buchman. And this question, I believe, is for you, Dr. Zummo, maybe for either. So there was a mention of 80% of patients in one, I believe it was in one of the phases of the trial, being choline deficient. Could you just talk a little bit about that data? There was a research study, I think, that was from the 90s, and someone's asking if there's anything more current.
Yeah, so the research study that one's from Alan. That was the phase 2, but then we conducted just a few years ago. I want to say it wrapped up in 2023, a global study where we had 88 patients who would meet the entry criteria for the registrational study around the world, similar sites as what we're running the study in today, where we tested choline levels using the assay that will be used in this trial. And what we found there was that approximately 80% or 78% to be exact were choline deficient. And we define choline deficiency the way that Steven Zeisel does, which is, less than 9.5 nanomoles per ml. The thing is that patients were actually much more deficient than that. And actually the majority of them were below 7.
Thank you. That's so interesting, especially, in this innovation is exciting to just know about the way to test and then a way to treat. So thank you. We have another question that just came in. Thanks, Jackie. Is there an opportunity to become a site for the trial?
Yes, absolutely. Please reach out to our clinical operations team. There is an email associated with that in this presentation. And yes, we're always looking for new sites.
Okay, perfect. Thank you. And anyone who is listening, if you didn't get information, maybe catch it on the slides. The recording will be shared. However, if you do have a question that we don't have time to answer, or if you didn't catch something from the slides, you can always email info, @oley.org, and we can make sure and connect you to the right resource. Okay, we have a question for Dr. Buchman. So it says, our team has looked into measuring choline levels, but do not have access to this lab. How do you measure, or are you assuming that most patients are deficient? I think, Jackie, you might have touched on that. And then there's a second part for those that do not have access to IV choline. Is taking oral doses able to help mitigate the effect of choline deficiency? And I could repeat, I know it's a 2-part.
But yeah, to answer the first part, there have been a couple assays that have been developed by companies. The problem is that there hadn't been a demand, because what would be the purpose of measuring choline in the blood if you can't do anything about it? So those will likely become commercially available, once there's a demand for it. There's a couple different lab companies. One company actually makes an automated machine that has been designed to measure it. And then in terms of the oral choline, that's the problem. As I mentioned, if you just take choline by mouth, you can do that, but if you really take it, it's not going to help much. The vast majority, over 70%, is going to be metabolized by bacteria that are present in your bowel. And those people that have short bowel syndrome are going to have more bacteria because normally the small bowel is close to sterile, not completely. But when the ileocecal valve is removed, bacteria come up from the colon and populate the small bowel. So you'll have even more bacteria and probably even more choline metabolism. And the problem is that it's metabolized to the trimethylamines, which is not a good thing. Again, the risk of heart disease, as well as the risk of strokes and other cardiovascular issues. So it's not, if you want to take a little bit, fine, but I don't think a little bit's really going to really help much, because even with the lecithin study, we gave 40 grams of lecithin. And the reason that I picked 40 grams is because that was the most that had been given in tardive dyskinesia studies without inducing nausea and vomiting. But that's only 19% choline. So people weren't getting that much. If you're going to take choline, I would probably recommend, rather than taking choline itself, to take it in the form of lecithin. But you're probably better off spending your money on a lobster or something, because it tastes better and it's cheaper.
So I think that, thank you, Dr. Buchman. Dr. Zummo, I believe this one would be for you regarding the trial. So given the findings of choline's role and importance in PN-supported patients for adults, do you anticipate this leading to adjustments with the neonatal and pediatric populations?
So I can tell you that we've had discussions with the FDA. It is definitely a patient group that they are very interested in us being able to understand the appropriate dosing regimen. What we've agreed with them is that we will try to, based on the PK data of our adolescent population, work with them to find a dose that goes down to neonates. We believe that it is likely that that patient population will require more choline than what we are testing. We do think that there is sophisticated modeling and simulation in the PK setting now that could allow us to get to that point of being able to understand dosing. We will work with the FDA. I can't say whether it will change, practice guidelines or whatever it was that was the question. But what I can say is that we are committed to trying to ensure that we know the appropriate dosing in that patient group.
Well, one of the issues is, and the reason that we have eligibility criteria in the study down to age 12 is because older children are really, as far as parental nutrition goes, are really basically like small adults.
Right.
But when you get smaller than that, the amounts required per body weight increase because choline is necessary for growth and development. And in the neonates and the newborns especially, where of course intestinal failure, liver disease is a huge problem in the preemies. The choline concentrations are as much as 200 in animals, 10 times what they are in adults, but they rapidly decrease within the first 30 days. And there have been several studies that have looked at choline concentrations in normals by myself, Tim Santongo, Steve Zeisel, and others. And so we have to determine what is the normal choline concentration for neonates and preterms, as well as in the younger children. So what are we going to be shooting for, and what dose is necessary? So for example, should the dose be based on weight? Should it be based on the energy? So for example, a neonate gets much more energy in terms of calories, for example, per body weight than an adult does, 100 maybe versus 25. And so the dosing is got to be sorted out. And then the other issue for neonates, where of course it is critical, and the FDA really has almost made us promise that we'll look at neonates, and is that how do you determine side effects, in a neonate, and how do you draw blood in a neonate, especially a preemie, where if you want to draw blood in the neonatal ICU before you even get to the incubator, the nurse is already swatting your hand. So, and they've got very little blood, and can we measure choline with, micro measures? So it's kind of complicated, but definitely that's clearly an unmet need that we want to work towards. But there's some more preliminary work that needs to be done.
Thank you, Dr. Buchman. And Dr. Buchman and Dr. Zummo, before we close, I'm going to pass to you each for any last words to wrap up. So I'll go to you first. Dr. Zummo, do you have anything that you would like to close with?
Just please, if you're interested in participating in our clinical trial, talk to your healthcare providers and reach out to us for any questions you might have. Thank
you. And Dr. Buchman?
I just want to thank Jackie and Protara for really stepping up to the plate here. Spending years actually even doing preliminary studies and raising money to do these things, because they're not cheap, and they were like a little company without money, and we went as far as we could, really. And I want to thank the FDA, not only for their active involvement with the current study, but they actually funded some of the work that I'd done earlier, as well as the NIH. So we're very thankful for that funding and that involvement, and I think everybody should be happy that Protara got involved. These are areas where there's so few patients that a lot of companies really have no interest.
Thank you. And thank you, Dr. Buchman and Dr. Zummo, for sharing your time, your clinical expertise with us. It was incredibly informative and exciting to know what innovation you've been working on. So thank you both very much. Thank you to all of you who attended and are listening to this live or on the recording. The recording is going to be made available on our website at oley.org/webinars. If you're not already, please make sure you're following Oley's social media channels, Facebook, Instagram, and LinkedIn, and getting our emails. You could always sign up at oley.org. So you could receive updates on upcoming events, including virtual and in person. We just had our annual national meeting last month in Charleston, South Carolina. And in the future, we have September 20th coming up in Kissimmee, Florida for another regional in-person meeting. And then on July 29th, next week at 7 p.m. Eastern Time, we have a virtual community connections for all living with nutrition support. And check out much more at oley.org or always info at oley.org. You can always email us at info@oley.org. All events are always free for those living with nutrition support, whether in person or virtual. And for all clinicians on the call, thank you so much for spreading the word about all of the great resources for your patients. And again, a big thank you, Dr. Alan Buchman, Dr. Jackie Zummo, and Protara Therapeutics for presenting and sponsoring this webinar. Have a wonderful day, everyone.